Hello everyone, welcome back to the class. In our previous class, we had started our discussion on acquisition of new genes and we had learnt about highly repetitive sequence in the DNA. So let's revise all the things what we had studied in our last class. So acquisition of new genes happens by four possible ways, that is by duplication and divergence of existing genes, or by acquiring genes from other species, or by gene fusion and fission, or by de novo gene origination. So through these four possible ways we can acquire a new gene. So we had discussed about genome and we defined the genome also. It is a sum total of all genetic material of an organism. It stores the biological information. So genome may be DNA or RNA. Genome can be classified into eukaryotic and prokaryotic. When we compare to eukaryotic, prokaryotic is simple, eukaryotic is complex. So to embrace the complexity of genome, there is a process of renaturation of DNA which helps in reading these sequence intensely. Eukaryotic genome consists of two distinct parts, that is nuclear genome. There is the DNA present inside the nucleus or the genome present inside the nucleus. Organellar genome is an example, like it is an extra nuclear that is present in the organelles, especially in mitochondria and chloroplast. And genome complexity can be studied, that is the total length of different sequences of DNA can be studied or can be measured through the renaturation kinetics of the denatured DNA. And these were the different types of genomes and their features. And we tried to differentiate between the renaturation and the denaturation. The renaturation and denaturation, to state or to define it simply, so there is a double-stranded DNA in between in front of you. So they have the complementary base pairing. If we subject this double-stranded DNA to heating, intense heating, the strands will break up and the base pairing between them will break. So this forms a single-stranded DNA. This is double-stranded DNA. This process of converting double-stranded DNA to single-stranded DNA by giving a heat, this process is known as denaturation. If consider we have broken single strands. After subjecting the double-stranded DNA to heating, we had got this single-stranded DNA. If we cool them, again they'll form a double-stranded DNA by complementary base pairing. It becomes double-stranded DNA. This process is known as renaturation. Means again they are rejoining, reannealing. This process is known as renaturation. So the process from denaturation, from denaturation to renaturation, where the base pairing is happening. This is the time where we can see the sequences of DNA. By renaturation of DNA, we can see many such sequences, genomic sequences inside the DNA. For example, so the DNA is having two types of sequence, that is non-repetitive and repetitive. the name, it suggests that non-repetitive means it is sequence is unique and there is only one copy and they undergo less rapid renaturation rate. They are present in the prokaryotes and lower eukaryotes. Repetitive suggests it is present more than one copy and they show more rapid renaturation rate. Eukaryotes have half of their DNA occupied in this sequence. Under repetitive, there are two types. Highly repetitive and moderately repetitive. Highly repetitive and moderately repetitive. Highly repetitive example we quoted yesterday, that is satellite. And for moderately we had quoted about transposable elements, that is transposable elements. So today we'll be learning about moderately repetitive sequence, as we had dealt with the highly repetitive sequence yesterday. So we'll be discussing about moderately repetitive sequence. See, yesterday we had gone through this highly repetitive sequence, that is satellite, where do this satellites are present and how we obtained by cesium chloride density gradient centrifugation, and where are these present in the centromere. Cesium chloride density gradient centrifugation, when we put the solution sample of the double stranded DNA, we'll get two bands, that is satellite band and main band. Majority of the genome is main band and having a small percent of the genome is satellite band, and they are present in many eukaryotes, and there are two types again in them, that is mini satellites and microsatellites, and this was the table to differentiate between each of them. So today we'll be learning about moderately repetitive sequence. See this moderately repetitive sequence example, transposable elements. So by the term there is one hint. Transposable means they can transpose. What do that mean? Transpose means they can shift from one side to another other side. They can move. Okay, so they can move from one side to another side, and being a moderately repetitive sequence, they have short sequence of DNA and they have the ability to move from one portion or one region of the genome to other. Based on this movement, this transposable elements are classified into two types, that is transposable element type 1 and type 2. Okay, based on the mechanism of movement and based on the on the basis of the self-sufficiency, they are further divided into other type, that is autonomous and non-autonomous. We'll discuss about this in future class. Let us consider this, that is type 1 and type 2 of the transposable elements. So to explain the difference between type 1 and type 2, let us begin with the DNA sequence. So consider there is a DNA, double-stranded DNA, in which some of the portion is having transposable element sequence or a moderately repetitive sequence, especially there is a transposable element in this. Okay. There will be a cut. So this this portion of the DNA which is having transposable elements is cut and it is transferred or transposed to another site, another DNA strand. So this is not it is very similar to cut and paste, right? The transposable elements is cut here in the donor DNA and it is transposed or pasted to the recipient DNA. This is donor DNA, this is recipient DNA. This is like a somewhat cut paste. So this type of transposable elements is known as transposons. That is type 2. Type 2. Cut and paste type of mechanism or movement is seen in transposon type 2. So there is a double-stranded DNA. There is transposable elements in between, and it will undergo transcription to form mRNA. After that, instead of translating to protein, it will undergo reverse transcription and it forms cDNA. Do we have transposable elements? Yes, transposable elements is intact here. After the formation of cDNA, the cDNA will complement to form other strand of DNA, that is, it becomes double-stranded DNA. Do we have transposable elements here? Yes, we have transposable elements here. This transposable elements is cut. It is copied. And it is transferred or transposed to the donor DNA. This is copied. It will remain intact in the double-stranded DNA. It will not move. It is just copying to the recipient DNA. Here I'll write copy. It will copy. Okay. So this type of movement of transposons or transposable elements can be seen in type 1, that is retrotransposon. Here it is copy paste. In the transposon type 2, that was cut and paste. So we'll go through it. So it is transposable elements is a short sequence of DNA. It have the ability to move in genome, and they make duplicate copies of themselves. The sequence is copied and inserted to the new site, and moderately repetitive sequence interspersed at multiple sites throughout the genome is transposable elements. Based on the type of mechanism of movement, we can classify transposable elements as transposon and retrotransposon. On the basis of self-sufficiency, we can classify transposable elements as autonomous and non-autonomous. So they are known as selfish DNA, jumping genes, insertion sequence, cassette or mobile elements. Transposition leads to mutation as it moves or the particular sequence move from one region of the genome to other region, so it takes away some of the sequence, so that will lead to mutation, which is a major source of genome diversity. It interrupts coding sequence and inactivates expression of genes. The first transposon was discovered in maize by Barbara McClintock in 1948. So these transposable elements contains transcription or translation termination signals which blocks the expression of other genes downstream. So based on mechanism of movement, as I said, there is transposon type 2, retrotransposon type 1. So here the type 2 is referring to cut and paste type of mechanism. Retrotransposon is copy and paste type of movement or mechanism. So first in retrotransposon, the DNA, if I have to systematically write, DNA is transcribed to mRNA. mRNA will reverse with the help of reverse transcription process. It forms cDNA, complementary DNA. Complementary DNA will duplicate, will form double-stranded DNA. This double-stranded DNA will have the transposable elements that is copied to the transposable elements, that is TE, is copied to the recipient DNA. So this is the procedure how the copy and paste happens in the retrotransposon type 1. If we have to discuss about the type 2, where the DNA, the double-stranded DNA is having the transposable elements in it, it is cut and it is pasted to the recipient DNA. So this is donor DNA, this is recipient DNA. That is example is type 2. Here you can see in detail. See the donor DNA with the transposon, the dark blue color or purplish color is the donor DNA with the transposon. The excision of that transposon takes place, and that excision will there is a removal of that dark blue color, right, from the recipient, from the donor DNA. It is transposed to the recipient DNA, which do not have that dark blue color here. Recipient DNA is void, so there the transposon is inserted. So the recipient DNA will now have the transposon, and the donor DNA, after loss of the transposon, they rejoin. So they rejoin to form a complete sequence. So this type is known as cut and paste. Here it is cut and it is pasted to the recipient DNA. This type we saw in type 2, and now we are discussing about type 1, that is retrotransposon. Here the donor DNA is having the retrotransposon, and it undergoes what? Transcription by the help of RNA polymerase enzyme. It forms a single stranded RNA. Then single stranded RNA, instead of translating, it undergoes reverse transcription to form single stranded cDNA, that is complementary DNA. This complementary DNA is converted to the double stranded DNA, which forms again the DNA. So in that there is retrotransposon present, and in the recipient DNA it is copied and it is pasted. It is copied and pasted. So in DNA, that is double stranded DNA, the transposon is present. In recipient DNA also it is present. So in both of them the transposon is present. It is not transposed completely. It is just copied to the recipient DNA. So these are the type of transposable elements based on mechanism of movement. See the class 1, short terminal inverted repeats, short flanking direct repeats at target site, transposase with the help of the gene transposase. By DNA intermediate, the transposition happens. Example IS1 in E. coli, TN3 in E. coli, AC, DC, DS, maize, P elements in Drosophila. Class 2, that is retrotransposon. It is long terminal direct repeats, short flanking direct repeats at the target site. With the help of reverse transcriptase enzyme, it undergoes reverse transcription. By RNA intermediate, the transposition happens. That is TY in yeast, copia in Drosophila, ALU in humans. So now we'll discuss about the other type of transposable elements based on the self-sufficiency. So the main difference between these two type of transposable elements is transposase enzyme. So I'll draw two type of transposable elements here. I'm drawing a transposable element. In this we have, or the transposable elements have a particular element or particular component. I'll draw another transposable elements. Which do not have this element. So there is empty thing. Okay. And I'll name it as transposase gene. So the element present here is transposase gene. The name suggests transposase genes means it codes for the transposase enzyme. What does this transposase enzyme do? Transposase enzyme aids in transposition. That's it. So the transposable element having this transposase gene is known as autonomous. So it has everything they need to promote their own transposition. That is the functional gene that encodes an active transposase enzyme and the terminal inverted repeat. The terminal inverted repeat, it means, so this has the boundary terminal inverted repeats. So inverted repeats. So along with the transposase gene, the AC element or the autonomous transposon is having terminal inverted repeat. What all do they have? Terminal inverted repeats. So now it is fully accomplished. It is fully having the elements needed for the transposition process. So it is having transposase genes, it is having terminal inverted repeats. That type of element is known as autonomous transposon, or example for that is AC element. In comparison to the autonomous transposons, non-autonomous transposons do not have any element like transposase gene. So they just have the inverted repeats, terminal inverted repeats, and they do not encode functional transposase as they do not have the transposase genes. Do they have transposase genes? No. They just have terminal inverted repeats. They need the assistance of this transposase gene which is present in the AC element to transpose. So they require transposase encoded by autonomous transposons for transposition. The transposon which do not have transposase gene and only have terminal inverted repeats, that type of transposon is known as non-autonomous transposon. And example is DS element. These AC and DS elements are found in maize. See here, AC element is having terminal inverted repeats along with the transposase gene. In DS element, there is discontinuous transposase genes. If there is a discontinuous sequence of transposase gene, it is considered that it is insufficient to produce or to synthesize the transposase enzyme. So that is the different DS elements have different deletions here. So the deletion of the particular sequence which codes for the transposase gene will lead to formation of DS elements. So there is no transposase genes. So that is the difference between autonomous transposon and non-autonomous transposon. Mechanism of transposition. So we had already discussed this in our previous slides. The conservative process is cut and paste mechanism, excision of sequence from its original position and reinsertion elsewhere, change in position of transposons in genome without increasing the copy number. It requires two elements, transposase and inverted repeats. The transposon using this mechanism are known as simple transposon. Remember, cut and paste mechanism was in type 2, that is transposon. So here the target site where there is a break and it is inserted. Then in replicative process, that is copy and paste mechanism, it creates a second copy. Original transposon remains in the place. Copy is inserted at the new position. Transposons using this mechanism are known as complex transposon. Here the target site is there. It gets copied and it is pasted. And you see here the original transposon remains in the place. So there is, we can say there is increase in the copy number, increase in the copy number. Copy was 1 and copy becomes 2 here. And based on the organism, transposable elements can also be classified into prokaryotic transposons and eukaryotic transposons. Prokaryotic transposons, that transposons are present in prokaryotic organisms, example IS elements. Eukaryotic transposons, where the transposons are present in the eukaryotic organisms, example retrotransposons, LTR and non-LTR, P elements in Drosophila, controlling elements in maize. So we'll study about each of these transposons in our next class. Thank you.